Hello everyone. Today my topic is membrane protein camouflage. According to a report released by WHO in 2015, Asia-Pacific region accounted for more than 62% of global deaths due to liver diseases. Orthotopic liver transplantation is the first-line treatment for end-stage liver diseases and acute liver failure, which have several drawbacks. Hepatocyte transplantation is an alternative to OLT, which has several advantages: less invasiveness to patients, wider sources of donor hepatocytes, and repetitive injection. However, it was also confronted with two main hurdles. First, limited sources of high-quality donor hepatocytes, and second, low cell engraftment and repopulation efficiency. So the causes of low engraftment efficiency are that about 70% of transplanted hepatocytes are cleared immediately after transplantation, and eventually only around 2% of hepatocytes can engraft into the liver plates. And this phenomenon is attributed to the activation of coagulation and complement system, and this process is termed IBMIR. Another cause is that loss of cell adhesion results in cell apoptosis during transplantation, which reduces the viable hepatocytes that is capable of engrafting the liver. So our previous work suggests that ROCK inhibitor, riposidil, inhibited MAC formation by stabilizing immunoregulatory membrane protein CD59A. Therefore, based on these data, we hypothesized that supplementing membrane proteins of hepatocytes enhances engraftment efficiency. Recently, platelet membrane has been used to encapsulate nanoparticles or stem cells to enhance their immunocompatibility and adhesive ability. Therefore, we decided to decorate hepatocytes with platelet nanovesicles, PNV, to supplement membrane proteins and promote engraftment efficiency. So for the design, in our project, we used human embryonic stem cells-derived hepatocytes and primary mice hepatocytes for the in vitro study. First, we acquired PNV by free-thawing and sonication, and fused PNVs with hepatocytes by PEG. The phenotypes of PNVs were also analyzed, including morphology, membrane protein expression, and size. For the detection of hepatocytes engraftment, we labeled hepatocytes with luciferase and two various fluorescence proteins. For bioluminescence imaging and competitive repopulation assay, respectively. To probe into the mechanisms of PNV promotion, we evaluated the complement activation by immunofluorescence staining and severe formation rate by suspension culture of hepatocytes. For the result, first, we successfully acquired PNV from mice whole blood and the Western blotting, suggesting that PNV expressed unique membrane proteins. After the fusion, the modified hepatocytes expressed membrane proteins enriched on PNV, suggesting the successful fusion of PNV and hepatocytes. Notably, PNV modification did not affect hepatocytes' viability. Next, we validate the pro-engraftment effect of PNV. According to bioluminescence imaging, we can observe that the luciferin intensity in mice liver was significantly increased, suggesting that the modification promoted hepatocyte engraftment. Considering the scarcity of platelet sources, we also examined the pro-engraftment effect of PNV, of long-term preserved PNVs. And we found that 6 months cryopreservation of PNV has a similar pro-engrafting effect as fresh PNV, suggesting that PNV can be stockpiled for a long time for the emergency use. In the competitive repopulation assay, the GFP labeled hepatocytes are as control, and the tomato-labeled hepatocytes were fused with PMVs. And we can observe that the tomato intensity in the mice liver was significantly higher than GFP intensity, suggesting that the modified hepatocytes have significant engraftment and prepopulation advantages over the control. For the mechanistic study, we found that the modification reduced the C5b deposition on hepatocytes, suggesting the modification inhibited complement activation and MAC formation. And since the cell adhesion molecules enriched on PMV, we also wondered whether it was also participated in improving the engrafting the liver, and we can observe that the modification enhanced the sphere formation rate on 3 days, suggesting the cell-to-cell interaction were enhanced, and also the ectopic engraftment was reduced, suggesting more hepatocytes retained in the liver. So in conclusion, PMV incorporates into hepatocytes membrane safely and efficiently, and the modification improves hepatocytes engraftment, and the underlying mechanisms rely on complement inhibition and cell adhesion enhancement. So our project validated that PMV modification promotes hepatocytes engraftment, and the modification conferred immunoregulation and cell adhesion properties on hepatocytes. And then PMV have low immunogenicity, high availability, and can be long-term preserved, making it pragmatic in clinical application. And finally, we established a novel experimental method called competitive repopulation assay, which can reduce experimental errors. Thank you for listening.